Hello students, I am Akshay Deshpande and welcome to the biology class. And this is the class 2 for the cell division and cell cycle, the chapter that comes in your first PUC. So till now, in this chapter that we have discussed is mainly about the interphase. We understood the importance of the interphase, what are the different stages in the interphase, and also we have understood what are the events that actually occurs in the interphase stage. Though the interphase is known to be a resting phase, but in fact it is the stage where the cell actually grows and accumulates various nutrients for some of the important events that actually occurs in the interphase. And one of that such important event that occurs in the interphase is we are having is the DNA synthesis, right? Also called as the S phase. So in this class, we will be what we can say focusing on the next phase of the interphase, that is mainly the M phase. Okay, and it is the M phase where we will mainly study about the mitosis. We will mainly study about the mitosis, and also we will try to understand why mitosis is a kind of important cell division when compared with the any other kind of cell division. Okay, the another kind of cell division we are having is the meiosis. Okay, that is another class, but however we have to quite concentrate on the significance of the mitosis. So let's start with the M phase. So now M phase, it is the phase, or it is the stage where you can see some of the dramatic events actually occurs. Okay, what do we mean by dramatic? Sudden changes. Okay, we do see a lot of sudden changes that can occur during the M phase. Okay, and we also refer the M phase as the equational division. Okay, now equational division in the sense where the DNA distribution, okay, DNA distribution, and also the number of chromosomes. Okay, the number of chromosomes are also equally distributed, that is what we mean by the term equational division. Okay, so now in the case of the M phase, we also see that there are two important events occur in the M phase, and that is the karyokinesis, and one more we are having is the cytokinesis. Okay, so now in the case of the karyokinesis, okay, we see that there is a division of the chromosome. Okay, it is the division of the chromosome. That is what we see in the case of the karyokinesis. Okay, the term karyo mainly stands for the nucleus. Okay, a nucleus, or ultimately the content that is present in the nucleus, that is the DNA. Okay, and the term cytokinesis, exactly as you can make out, the cyto mainly stands for the cytoplasm. Hence we can say that it is the division of cytoplasm. It is mainly the division of cytoplasm. Hence it is called as cytokinesis. So these are the two important events that actually occur in your M phase. But though we divide the cases, or divide the M phase into karyokinesis and into cytokinesis, we observe that these events are progressive. Okay, now what do you mean by progressive? It means that after the karyokinesis you see cytokinesis, and after cytokinesis again the cell grows, and after the cell grows it enters into the M phase, and the starting of the M phase you see there is a karyokinesis and again cytokinesis. Okay, so this is it is called as the progressive what we can say stages. They are serially occurring events. But interestingly, we also see that this kind of categorization is mainly meant for our study. Okay, we say karyokinesis and cytokinesis, and this categorization is mainly meant for our study, but that is never the case in actual process. Okay, so that is what we say over here. There is no clear-cut distinctive differentiation between the events that actually are occurring in the M phase. There is no such clear-cut distinction. It just goes on occurring. That's it. Okay, we have to remember this point. So M phase is mainly we observe that there is an equal distribution of the chromosomes. Okay, and at the end of this particular result, that is after the distribution of the chromosomes, you see that the daughter cells, okay, the daughter cells remain, okay, they remain diploid only. Right? Yes, they remain diploid. It means that if you are having a cell over here, okay, and if this cell is diploid, the daughter cells, what you get, okay, the daughter cells, these two are the daughter cells. Even the ploidy condition of these two daughter cells are going to be 2n in condition. Okay, 2n in condition. So this is what we mean by the equational division. And as also we mentioned that you find karyokinesis and cytokinesis as two important stages in the M phase, and all the events are progressive in nature. None of these events are having any clear-cut distinction in between them. Okay, it is we who have identified certain changes that occurs during certain parts of the cell division or certain time of the M phase or the mitotic phase. So now the karyokinesis is actually divisible into the prophase, okay, metaphase, and next one we are having is the anaphase and the telophase. Okay, these are some of the important what we can say stages of the karyokinesis. Okay, the first one we are having is the prophase, next one we are having is the metaphase, the third is the anaphase and the telophase. Right, so you people do have a very fairly good idea regarding what these phases actually do. Okay, so now if we have to speak about the prophase, you see that there is a unwinding and condensation of the DNA. Okay, so now here we can draw the pictorial representation of a cell that is actually present during the prophase stage. Okay, so when the cell is actually before entering the prophase stage, the chromatin fibers is randomly distributed throughout the cell. Okay, so now this is called as the nucleus. Right, so now as soon as the cell enters into the prophase, you see that there is what you can say disappearance or initiation of the disappearance of the nuclear envelope. Okay, this is the nuclear, okay, it is the nuclear envelope. Okay, and the nuclear envelope starts to disintegrate. Okay, it starts to disintegrate, or in other terms starts to break. It actually starts to break. Right, so we can say that a nuclear envelope actually starts to disappear or starts to break. Now as the nuclear envelope starts to break, even the elements that are associated with the nucleus, okay, even the organelle that is associated with the nucleus, okay, that is mainly the endoplasmic reticulum. Okay, it is mainly the endoplasmic reticulum. Now the endoplasmic reticulum is mainly attached with the nuclear envelope, right? Even the endoplasmic reticulum also starts to disappear. Okay, even we can see that even this endoplasmic reticulum also disappears. And you see that there is a initiation of condensation of DNA. Okay, DNA condensation occurs. okay, DNA condensation occurs, right? So you can see that these thread-like structures, which is called as the chromatin, starts to condense, okay, starts to condense to form a thick thread-like structures, okay, a thick thread-like structures. Yes, this is how what we can say the chromatin fibers starts to appear in the nucleus over here, okay, and this process is called as the DNA condensation. Now condensation mainly means it is a compaction of the DNA, okay. So what is the ultimate result that you see after the DNA condensation? The DNA is condensed into form a structure called as the chromosome, okay. It is what? Formation of the chromosome. So now, if you have to look into the chromosome, we all know that the chromosome is actually having what we can say two chromatids at each attached to the one centromere, okay. Now this structure is called as the centromere, and this structure is called as the chromatid, okay. It is called as the chromatid. Right now, what happens is that the centromere also develops one structure above its region, okay, and this thick cup-like structure, okay, is called as the kinetochore. Okay, it is mainly the kinetochore. Now, this kinetochore is a structure where, or it is the site, okay, it is the site or point of attachment of the spindle fibers, okay. It is the site or the point of attachment for the spindle fibers. So now the spindle fibers are mainly made up of microtubules, okay. It is mainly made up of the microtubules. If you have to recall about the microtubules are actually part of the cell cytoskeletal elements, okay. They are part of the cell cytoskeletal elements. The cytoskeleton mainly comprises of the microtubules, microfilaments, and also the intermediate filaments, right? So here the microtubules are mainly involved in a what we can say elongated thread-like structure called as the spindle fibers, which ultimately attach to the kinetochore. So we can say that the spindle fibers are not attached to the centromere directly. Instead, they are attached to one particular structure called as the kinetochore, which are present above the surface of the centromere, okay. So he can see over here that there is a disappearance of the nuclear membrane and other organelles. Also, we can see that the duplication of the centrosome has occurred during the S phase. In some cases, some textbooks also mention that the centrosomes actually duplicate during the prophase only, okay. But however, in general context, okay, more accurately speaking, we can say that the centrosomes have actually duplicated during the S phase itself, okay. So we also know that S phase is the stage where the DNA synthesis or DNA duplication actually occurs, right? So along with the DNA duplication, you also see that even the centrosome also duplicates. So now the centrosome that has duplicated also moves towards the opposite poles, okay? So here what happens is that this is called as the centrosome, okay, centrosome, and this is another centrosome, okay. Here we are having one more centrosome, okay. So this is one centrosome and this is another centrosome, which you can see that they have now started to move towards the opposite pole. So this is one end and this is another end, right? So opposite ends are generally called as the poles. Now, it is mainly from the centrosomes that are involved in the formation of asters. Now, asters are important for maintaining, okay, maintaining the stability. They mainly involved in the stability of spindle fibers, Okay, they're required for the maintenance of a proper stability in the case of spindle fibers. That is what they are observed, right? But however, the asters are not the structures that finally attach to the kinetochore. Okay, they are required for mainly the stability part of the spindle fibers. Okay, but however, along with the asters, one more structure, what happens, are observed, that is called as the spindle fibers, and these spindle fibers are mainly attached to your what we can say kinetochore of the chromosome. Okay, so if you consider aster and spindle fibers, both of them together, they mainly form the apparatus which is called as the mitotic apparatus. Okay, now this mitotic apparatus mainly is involved in what we can say attachment to the kinetochore of the chromosome, and it mainly brings or splits the chromosome into two. Okay, but however, you can see that this chromosome is only one in nature. Okay, that is a different part of the event that you people will be shortly studying now. Now, next one. The next phase we are having is the metaphase stage, right? So now what happens in the metaphase stage is that if you have to draw it in a more clear way, you see that the chromosomes are arranged interestingly in a state, okay, in one particular part, okay, or we can say that they are attached or arranged in only one way. So you can see that this plane, okay, this plane is called as equatorial plane, okay, or we can even say that it is called as the metaphase plate. It is called as the metaphase plate, right? So now what happens is that the chromosomes have condensed to the most what we can say compact part. Okay, the chromosomes are clearly visible in the metaphase. Okay, so the karyotyping, okay, the karyotyping, right? You people will be studying about the karyotyping in the part that is the principles of inheritance and variation, and also in the what we can say even in the reproductive health and all. We know that the karyotyping is mainly used to determine the sex chromosomes of the fetus. Okay, but however, the main intention of the karyotyping is mainly to determine the disorders, genetic disorders, right? Amniocentesis, mainly what I'm trying to refer to. Okay, so the chromosome photographing, that is the formation of chromosome, is very much clear at which stage? It is mainly during the metaphase stage, right? And interestingly, you also see that the nuclear envelope has completely disappeared. The nuclear envelope has completely disappeared, and there is a complete condensation of the chromosome. The most compact form of the chromosomes are observed during the metaphase plate. Okay, so hence the study of chromosomes, okay, generally the study of chromosomes are more clearly observed in the metaphase. Right? Yes. Now, as I said that you see that at the centromere there is a formation of a structure called as the kinetochore. Okay, and the kinetochore is more clearly visible. Okay, it is more clearly visible during what we can say metaphase stage. Okay, now the arrangement of these what we can say chromosomes in the equatorial plane is called as the metaphase. And you can also see that the spindle fibers that have arose from the centrioles have attached now to the kinetochore. So now, yes. So these are the asters what we are speaking of. The dark line what I have drawn, they are actually the asters over here, right? Yes. So now this is the centrosome or centriole. Okay, it is mainly the centriole or the centrosome. And now the spindle fibers have attached to the kinetochore. So you can see that the chromosomes are completely hanging in between or at the equatorial plate. And this kind of arrangement we call it as the metaphase plate, right? So here, that is what the points have been mentioned. The chromosome arrangement in the equatorial region is called as the metaphase plate, and the spindle fibers are attached to the kinetochore. So these are some of the salient features of the metaphase part of the M phase. The next one we are having is the anaphase. So now in the case of anaphase, you see that the chromosomes start to split. Okay, so this is the what we can say anaphase, and yes, this is so this is one chromosome, and this is another chromosome. Okay, yes, another chromosome, and here we are having the centrioles. Here we are having the centrioles attached over here, right? Yes. So this is how the diagrammatically we can show it over here, right? So now these sister chromatids will actually, the chromosome will actually split now, okay, and they are pulled towards the opposite end. Okay, so one part will move to this end and the another part of the chromosome is moving to the opposite end, right? So you can see that the chromosome actually splits. So now what is mainly responsible for splitting of the chromosome? Okay, it is said that the pulling effect, okay, pulling effect of the what we can, it's not centriole, sorry, we can erase this. Okay, it is not the centriole, it is mainly what we can say the pulling effect of the spindle fibers. It is mainly pulling effect of the spindle fibers. Okay, now why does or we can say how does the pulling effect is achieved? Okay, the pulling effect is achieved by shortening, okay, shortening of spindle fibers. Okay, so what happens is that there is always a constant elongation and shortening of the what we can say spindle fibers. So now as soon as the spindle fibers get in contact or they establish the contact with the what we can say kinetochore of the chromosome, you can see that the spindle fibers will start to shorten in length. The shortening of the spindle fibers is in turn resulting in an effect called as the pulling effect, and the pulling effect finally results in the splitting of the chromosomes. So now the chromosome that has actually split, the sister chromatids are pulled towards the opposite end of the poles. And now you also see that the pulling action of the spindle fibers is mainly responsible for the separation of the sister chromatids. So in a nutshell, if I have to tell what are the important events that can occur in the anaphase, it is mainly the splitting of the sister chromatids, okay, and moving of these sister chromatids towards the opposite end. So now in the diagram you can see that when it is being pulled, the what we can say the centromere is actually being dragged, while you can see that arm of the chromosome is pointing forward. Okay, it is pointing in this particular forward direction. Yes. So in some cases the chromosome can look like a J-shaped, okay, or it can look like I-shaped or L-shaped, okay. So that is mainly because of the length of the chromatids that are attached to the centromere. So we even classify the chromosome based upon the what we can say what kind of appearance it is observed during the anaphase stage. That is also observed. The next stage we are having is the telophase. Now telophase is the final stage of what we can say karyokinesis. Okay, and in the case of the telophase, you see that the chromosomes have actually reached to the end of the poles, right? So if I have to show this diagrammatically, you can see that the what we can say, this is one pole, okay, and the chromosomes, what we can say, reappearance of your nuclear envelope. Okay, yes. So you can see that the chromosomes have actually reached to the end poles. Okay, end poles, completely reached to the one end of the pole, right? And there is a decondensation of chromosomes, right? They lose their identity as chromosomes. Instead, they become decondensed and become into chromatin. They actually become into what? They become into chromatin. Now, this process is called as the decondensation. And finally, you also see that there is a reappearance of the nuclear envelope and also the remaining cell organelles. Okay. Now, what are the remaining cell organelles? Exactly the cell organelles that had disappeared earlier in the prophase. That is mainly the endoplasmic reticulum, Golgi apparatus, and also the lysosomes and etc. Okay. But what about the plastids and mitochondria? Okay, what about the plastids and mitochondria? Now you have to remember that the mitochondria and plastids, they actually don't disappear during the mitosis. Okay, instead you see that the mitochondria, if the mitochondria or the plastids that are present to the nearest pole, they become the part of that particular cell. They mainly become the part of that particular cell. Now, the last stage we are having is the cytokinesis. Okay. Now, cytokinesis, as we all know, it is mainly referring with the term that is the division of the cytoplasm. Okay. Now, the division of the cytoplasm can be equal in some cases, or it can be unequal in some cases. Okay. In the case of oogenesis, okay, oogenesis, okay, you see that the division of cytoplasm. The division of cytoplasm is unequal. It is mainly unequal in nature. So we can say that, of course, in the case of oogenesis, the type of cell division is different. That is mainly it is meiosis. But that doesn't mean that there is no cytokinesis occurring. Even in the case of meiosis, the cytokinesis occurs. But in general terms, we can say that the cytokinesis is equational in nature. Okay. But however, in oogenesis, the cytokinesis can be unequal in division of cytoplasm. Okay. Now, in the case of animal cells, you see that it is mainly achieved through the cleavage furrow. And in the case of plant cells, it is mainly with the help of the cell plate, right? So now, here in the case of animal cells, what happens is that a small pit-like structure actually appears. A small pit-like structure actually appears. Okay. Now, this is the nucleus. And this pit-like structure is called as the furrow. And the furrow starts to deepen gradually. Okay. And finally, this two part will reach, the furrow will actually reach near to each other and pinch off, resulting in the formation of two daughter cells. Okay. It results in the formation of two daughter cells. But in the case of plant cells, we mentioned about the cell plate. It is mainly because you see that the plant cells are quite rigid in nature because of one important reason, that is the presence of the cell wall. Okay, the cell wall is quite rigid in nature. It's a rigid structure. As a result, you see that there is no contraction of the, what we can say, plasma membrane, and that results in the formation of a cleavage furrow. But you see that there is another way, that is a formation of a small plate-like structure called as this structure is called as the cell plate. Okay, cell plate. And this cell plate gradually increases in its length and finally dividing the two cells. finally dividing it into what? Two cells. In fact, this cell plate represents the middle lamella. Okay, it actually represents the middle lamella. Okay, and this middle lamella is acting as a glue-like structure that is ultimately present between the walls of the plant cells. Okay, now in some cases you see that soon after telophase, you see that there are two nucleus. There are two nucleus present in a single cell. Okay, and what happens is that there is no cytokinesis immediately occurring. Okay, so we can say that in a single cell there is more than one nucleus, or there are multiple nuclei present in a single cell. Okay, and such kind of a state is called as the syncytium. Okay, now syncytium is a condition where more than one nuclei or multiple nuclei is present in a single cell, right? And that is generally observed in the case of a liquid endosperm of the coconut. If you open the tender coconut, you can see water. Okay, that water is none other than the liquid endosperm. Okay, and what we can say, the white fruit or the white pulp that is present inside that coconut is actually the cellular endosperm that you observe. So now we can look into the significance of mitosis. What is the role that the mitosis actually plays? The mitosis is required for one of the reason that it mainly maintains the genetic content. Okay, so as we mentioned earlier, that it is called as the equational. It is called as the equational division. Okay, it is called as the equational division. Now it is called as equational division because ultimately at the end of cell division, you see that the mother cell, okay, the mother cell was diploid, right? The mother cell was mainly diploid, and even the daughter cell is also diploid in nature. It is also what? Diploid in nature. So hence we can say that there is no reduction in the what we can say in the amount of DNA or number of chromosomes. Okay, even the mother cell was diploid. Similarly, even the daughter cell is also diploid. Okay, so the genetic ploidy condition, the number of what we can say chromosomes is maintained, and also the genetic content is also maintained. Hence we say that it is a kind of equational division. Now, the growth in the case of multicellular organisms is ultimately achieved because of mitosis. Okay, you can see that bacteria is actually what we can say, it is a unicellular organism. Okay, and mitosis is actually what? Acting as a reproductive mechanism. Okay, it is actually a mode of reproduction. It is actually a mode of reproduction in the case of the organisms such as the bacteria. But in the case of multicellular organisms, you see that the mitosis is required for the growth. The growth is ultimately achieved in the case of multicellular organisms with the help of mitosis. And because of mitosis, what happens is that there is a division of a nucleus, and also there is a division of the cytoplasm. Okay, so in every cell there is a respective ratio of a nucleus and also the cytoplasm, and this proper ratio between the cytoplasm and the nucleus is called as the nucleocytoplasmic ratio, and ultimately the maintenance of the nucleocytoplasmic ratio is achieved by the mitosis. Okay, it is mainly through mitosis you see that the nucleocytoplasmic ratio is achieved or maintained. Okay, but however, the most important significance of the mitosis is mainly the cell repair mechanisms. Okay, so the replenishment of the dead cells. Okay, replenishment of the dead cells. For example, our skin epidermis, upper layer of the epidermis of our skin. Now similarly, even the lining of the gut, constantly our stomach or our intestine, the villi will be having surface, that is the villi will be having epithelial cells. These epithelial cells are constantly, what we can say, they are removed during the digestion process. So now these epithelial cells present in the gut lining are regenerated. Okay, they are reformed, and that is again mainly because of the mitosis. Even the blood cells, as we all know, the RBC is having a lifespan of around 120 days. So the dead RBC cells are actually destroyed in the spleen, but the new blood cells are produced immediately through the bone marrow. And even in the case of plant cells, the lateral meristems, that is the lateral cambium, and also the apical meristems, that is the root meristems, and also the apical meristems, they are mainly required for the constant growth of the plants. That is why plants are constantly growing throughout their lifetime. Okay, so hence we can say that we have discussed some of the important aspects of the mitosis, that is the M phase, and also we looked into the significance of the mitosis. Now we can start with a few questions for our discussion. Okay, the question is, at the end of the mitosis, the content or the volume of the DNA is 2C, 3C, 4C, 6C. So you know that the content of is mainly increased during the S phase. But however, we can say that during the mitosis and at the end of mitosis, the volume is reduced back to its original content. Okay, and that is 2C only. So hence the right answer for this question we are having is the 2C. Next one, the chromosome number during the mitosis, during and after mitosis is increased, decreased, remains the same, all of the above. We know it is remains the same. Okay, even during the mitosis and after the mitosis, you see that so if I have to show this in a diagram, let us assume that we are having one chromosome over here, right? And this is one cell, and after division you can see that the sister chromatids splits and it enters in one cell, and you can see that the another chromosome enters into another pole and becomes into the part of the another cell, right? So you can see that during the mitosis and also after the mitosis, or even before the mitosis, even the before mitosis actually has started, again you can see that the mother cell actually had one chromosome, and even the daughter cells are having one and one respective chromosomes, right? So in this particular way we can say that the number of chromosomes are going to remain same. So the next question, which of the following statement about the mitosis is correct? The genetic content is doubled in the M phase, and the number of chromosomes doesn't increase, and the division of cytoplasm is called as the karyokinesis. The condensation of DNA is the anaphase. Okay, the condensation of DNA occurs mainly in the starts in the prophase. Okay, but however becomes more what we can say more clearly visible in the case of metaphase. So this is not a correct statement, and the division of cytoplasm is actually called as the cytokinesis, not karyokinesis, and the chromosome number does not increase, and this statement is actually the correct statement. The genetic content is doubled in the S phase, not in the M phase. So B is the right answer for this particular question. The next question, let me erase this. Yeah, the next question, which of the following event is a characteristic of prophase? Splitting of sister chromatids, a disappearance of the nucleolus, arrangement of chromosome at the metaphase plate, all of the above, right? So you can see that splitting of sister chromatids actually occurs in the anaphase, but the disappearance of the nucleus is actually the characteristic of the prophase. So the B is the right answer over here. And arrangement of the chromosomes of the metaphase plate occurs in the metaphase stage, not in the prophase. Next one, and the duplication of centrosome is observed in the G1 phase, M phase, S phase, and G2 phase. It is mainly observed in the S phase. So C is our right answer. The next one, the chromosome in the metaphase are equatorial plane at the poles, decondensed state or unwinded state. It is mainly at the what we can say equatorial plane, and that plane of arrangement in the equatorial region is called as the metaphase plate. So A is the right answer for this question. The next is the chromosomes with the sister chromatids are visible at the anaphase, metaphase, telophase, and prophase. We know that they are more clearly visible during the metaphase time. So hence B is going to be our right answer. The next one, the attachment of the spindle fibers to the kinetochore is observed in metaphase, telophase, prophase, anaphase, right? It is mainly in the metaphase time. Okay, completely attachment is observed during the metaphase moment or phase of the mitosis. The next one, which of the following statement is not true? In prophase, chromosome splits, okay, and in metaphase, chromosome arrangement is in the metaphase plate. The centrosome is duplicated in the S phase, and Golgi complex reappears in the telophase. You see that it is actually true. D is true. C is also true. Even B is also true, but the A is the not true statement, and we can say A is our right answer. Okay, the splitting of chromosome occurs in the anaphase stage, not in the prophase. The next one, the structure that disappears in the prophase is endomembrane system, nucleolus, nuclear envelope, all of the above. So we know that endomembrane system mainly comprises of the what we can say endoplasmic reticulum, even the Golgi apparatus, and even the lysosomes, and also the vacuoles and all. All of their part of the endomembrane system. So endomembrane system along with the nucleolus and even the nuclear envelope, all the three structures disappear. So we can say that D is the right answer, all of the above. The difference between the meiosis and mitosis is duplication of DNA and the cell division rounds, sister chromatid separation, and all of the above. So you can see that duplication of DNA occurs between both the meiosis and also mitosis. Hence we can say that it is a common feature. Cell division rounds, it is actually observed again both in meiosis and mitosis, but the cell division is only occurring one time, but the cell division in the meiosis is occurring two times. Hence this is the main difference between the what we can say mitosis and meiosis. So B is the right answer. Sister chromatid separation occurs both in mitosis and also in meiosis. Okay, in meiosis too there is sister chromatid separation, and in mitosis also there is a sister chromatid separation. The next question, which of the following event does not occur in the prophase? A disappearance of nuclear envelope and disappearance of endomembrane system, centrosome duplication, initiation of chromatin condensation. Okay, so you can see that centrosome duplication has already occurred in the which stage? Already it has occurred in the S stage or S phase of the interphase of cell cycle. So C is going to be not the event that is occurring in the prophase. The next one, sister chromatids is the separation of sister chromatids is a characteristic of the telophase, anaphase, prophase, metaphase. We know separation of the sister chromatids occurs at the anaphase, hence B is our right answer. The next question, in animal cells, cytokinesis involves separation of sister chromatids, contraction of microfilaments, depolarization of the what we can say microtubules, none of the above. Okay, it is mainly the contraction of the microfilaments. Okay, the cleavage, okay, cleavage furrow, what we have seen during the cytokinesis. Okay, the pit-like structure that is mainly formed, okay, this is called as cleavage furrow. The cleavage furrow starts to deepen gradually is mainly because of the contraction of the microfilaments. And what are the microfilaments mainly made up of? Microfilaments are mainly made up of the actin molecules, mainly the actin molecules. So B is going to be our right answer over here. The next question: how many chromosomes will be present during the G1, S, and M phase respectively if it has 14 chromosomes at the interphase or at the beginning, right? So there is no increase in the number of chromosome, and also there is no decrease in the number of chromosome. It's going to be remaining same. So we can say that the number of chromosome that is going to remain same after or during the G1, S, and M phase is 14, 14, 14, and 14. So B is our right answer. Okay, as I had said earlier in the beginning of the class, during the all the stages, the number of chromosome are going to remain same in the mitosis. Yes, remember that point. The next question: Golgi apparatus, nucleolus, and endoplasmic reticulum reform in telophase, anaphase, metaphase, and prophase. We all know they come back, they reappear back in the telophase. Hence, A is going to be our right answer. The next question: which of the following is not a significance of mitosis? It is cell repair, cell growth, and restoring of the nucleocytoplasmic ratio, gamete production. No, gamete production is not a part of mitosis. It is actually significance or importance of the meiosis. So D is going to be our right answer for this question. The next one: in the plant cells, cytokinesis occurs through furrow, contraction of actin filaments, and the cell plate formation, all of the above. In the case of plant cells, it is mainly through the cell plate formation. Hence, C is the right answer over here. Next one: the cell plate represents the walls of a plant cell is the plasmodesmata, middle lamella, protoplast, and cell membrane. It mainly represents the middle lamella. So B is the right answer over here. The next question: after the cytokinesis, the cytoplasm does not divide immediately and has multiple nucleus in a single cell. Now this cell is referred as exomitosis, the mitosis, or soma clone, or syncytium. So we know it is actually the syncytium which is having multiple nuclei in a single cell. So this is mainly about the M phase. This was a very short class because there is no much detailed explanation that is required in this particular part. Okay, so in the next class we will mainly meet and discuss about the meiosis. Okay, and that is going to be the last class for your the cell division chapter. Okay, and we will also try to look into some previously appeared questions in that particular class only. Thank you.